All right, just so that I'm respectful of everyone's time, I wanted to welcome everyone to the next edition of the USC-Buck Nathan Schock Center Journal Club. Today we have the privilege of hearing from Max Thorwald, who's a ridiculously talented postdoctoral fellow in the Finch Lab here at the Davis School of Gerontology at USC. And he's truly an expert in ApoE and its effects on the brain. And so this is actually a really perfect paper for him to be leading the discussion on. I think it's probably easiest if we save questions for the end. That said, I'll monitor the chat in case there's something really urgent that you wanted to have addressed, but there'll be time at the end for a Q&A. So if you can save your questions till then, I think it'll make it a little bit smoother. But with that, take it away, Max.
Okay. Hi, Max Thorwald. I work for Caleb Finch. Today, I will be presenting Destabilizing Heterochromatin by ApoE Mediates Senescence. This is a Nature Aging paper from 2021. And so they're interested in looking at the role of nuclear ApoE, which is something I'm also interested in. So some background on ApoE. So apolipoprotein, it's the primary cholesterol shuttle within the brain. And there are three main isoforms, E2, E3, and E4. You inherit one copy from mom and one from dad. So you can be a 2-3, a 3-4, or 4-4, et cetera. So 80% of people are ApoE3-3 carriers. Roughly 10 to 15 are ApoE4. And only 5 to 7% are ApoE2 carriers. And structurally, E2 and E3 are very similar. There's a difference of a cysteine in place of an arginine at the 158th amino acid for E2. And the E4 has two arginines. And you can see that they're structurally very different. They carry different amounts of lipids. And so they're involved in several disease states, particularly amyloid and Alzheimer's disease. And so ApoE4 is the largest heritable risk factor for the development of late onset Alzheimer's disease. So ApoE is involved in amyloid clearance. There is a potential difference in binding to amyloid and removal of it. And it is also associated with other pathologies such as increased cardiovascular risk. So the E2 allele is actually protective for Alzheimer's disease, but for heart disease, the E2 and E4 allele are not. And then there's some literature on cancer and also on these ApoE isoforms. So some other interesting things, actually I'm gonna go back for just a second. The E2 allele is the most common, or not most common, most recent, excuse me. The E4 is the ancestral allele. And then as it's evolved, it's now gone to E2. So those were some basic roles of ApoE and things that are commonly studied for ApoE, but there are currently only 8 papers in the literature identifying ApoE in the nucleus. Many of these papers were in the late 90s and early 2000s across a wide variety of cells. So on the left, these are Chinese hamster ovary cells where they've stuck GFP on ApoE, and they have found that ApoE actually localizes to the nucleus in these cells. And they also did some experiments where they starved them, and by starving them, they saw that there was an increase in nuclear ApoE. Not much more was said from that. No functionality, no rationale or why that was happening. This is another paper more recently in 2017, which looked at ApoE in an AD brain and found that it was co-localized to the nucleus, particularly in microglia cells. So there have been a few other papers that have popped up, but nothing really cementing the role of nuclear ApoE. Then in 2016, there was a paper from Rauman Rao, Thindakara at Buck, actually. And so what they did was chip sequencing of an E3 and an E4 cell line. So they took A172 glioblastoma cells, they transiently transfected them with E3 or E4, they were homozygous. And so what they found was that there were 1,700 promoters in the E4 data set that were not found in the E3 data set. And one of those promoters was SIRT1. Well, it binds to both, but they targeted SIRT1. And so they did some more assays and they found a localization sequence within the SIRT1 promoter. And then they showed using luciferase assays that if you put ApoE3 basically in a tube with that sequence, you get some binding. And so they claim eventually that SIRT1's promoter has higher binding affinity with the E4 rather than the E3, and that this may explain some of the differences during Alzheimer's disease. So on the left, this is a schematic from one of their later papers. Again, there's the 1700 promoters. This is a proposition they were making for ApoE. However, with this, not much else has been done since then, and still no true identification of what ApoE is doing. So the current paper, they even put in their introduction that they're aware of these papers, and they too are interested in potentially what's going on with ApoE and aging. And so what they find, their main findings, is that ApoE drives senescence in mesenchymal progenitor cells. And if you knock out ApoE in these cells become resistant. And then they put at the very end, after a lot of lengthy experiments, that they believe that ApoE interacts with a protein called CAP1 on the nuclear envelope and may facilitate lysosomal degradation of heterochromatin. So the very first thing they did was they got primary human MPC cells. So if you can see the ages up here, 16, 23, 28. So these are their young primary cells. And then some old ones, 76, 79, and 80. And clearly you can see that there's an increase in ApoE levels. And this is from a whole cell lysate. And so their very first assertion is that the elderly, at least for MPCs, have higher ApoE levels. So maybe ApoE is associated with senescence. Then what they did was a cellular model of aging, which is essentially late passaging cells. So here we have replicative senescent human MPCs here. And so with 3 passages, that's their baseline. And then they have another set at 9 passages. And you can see that there's an increase in ApoE content. So then they took 2 models of accelerated aging. So Hutchinson-Gilford progeria and Werner syndrome, which is also technically a type of progeria. And they compared them to wild type cells. And they found that there was a large increase in ApoE as well. So this further supported their notion that maybe there was an increase in ApoE with aging. Here's some more background data from those same cells. So they included some qPCR results for CDKN1A and ApoE. So CDKN1A is associated with senescence, and they show in the late passage it goes up, so does ApoE, and then the HGPS cells, the same thing, and also the Werner cells. So next they wanted to know what would happen if they knocked out ApoE. So they took embryonic stem cells and they CRISPR'd ApoE out of one line, and then they transformed them using conditioned media for 30 days to get them back to being MPCs. And so what was interesting is that they found that there's essentially no difference in cell cycle or localization of Ki67, senescence associated marker for these cells. And they also looked at OCT4, which is involved in stem cell proliferation, and they looked at it for methylation, and there was no difference in methylation. So they make the statement that ApoE seems to be dispensable for ESCs. So here's where things start to get more interesting. So by simply knocking out ApoE, you see an increase in proliferation by passage number, and then clonal expansion over here as well. And so these are the wild type cells, and they went all the way to passage 12, but we get 3 more passages with the ApoE knockout. And see, there's a quite big increase in clonal expansion with knocking out So some more background on those progeria models. HDPS in particular is marked by a mutation in the lamin gene. So they were curious what would happen if to lamin essentially in these knocked out cells. And so they found that ApoE was, well, actually the relative intensity of LAP2, lamin-associated protein 2, increased in the ApoE knockout cells. And so on the right is actually some RNA-seq data. And so these are their control cells. And they show that lamin B receptor 1 actually goes up when you knock out ApoE. And then 3 markers of senescence go down, CDKN2A and interleukin 6 and IL-8. So far, pretty strong evidence for ApoE being associated with aging. So next, what they did was mass spectrometry using an ApoE IP, an immunoprecipitation. So they took lysates, and then they took an antibody specific for ApoE. They pulled ApoE out of that lysate, and then they mass spec'd it. And then they sorted those proteins by chromosome interactors and also nuclear envelope proteins. And so I'm not certain why they didn't spend too much time on the chromosome interactions, because these are actually pretty interesting to me, but they stuck with the nuclear envelope story over here. And so what they found were several proteins, of course, in the nuclear envelope, such as emerin and lamin B receptor. And so here's the confirmation data over here in the knocked out cells. Their IP Western blot shows that there's no association between ApoE and CAP1, another envelope protein, lamin B receptor, and emerin. Down here is their positive control. So we pull out ApoE, probe for it, and we see a signal. So they know it's working. So the next thing they did was they made flag-tagged ApoE with kidney cells, and then they stuck that flag-tagged ApoE back into the knocked out ApoE MPCs. And so the flag tag allowed them to identify where ApoE was more readily, more easily. And they see that it is actually co-localized with emerin along the nuclear envelope. So this is 2 micrometers. And so when you merge them, you can see that they overlap. And this is a densitometry image of that. So next they used the same flag tagged cells, but they stuck a nuclear localization signal on this ApoE flag tag. And sure enough, you see much more localization of ApoE to the nuclear envelope. And then when putting a nuclear export signal there, it starts to leave. So you can see that here with the histogram, there's an increase in ApoE to the nuclear envelope, and then a decrease with the export signal. Over here, we have Western blotting for CAP1, lamin B receptor, and emerin, the 3 that they're going to work with for the rest of this paper. And so the luciferase flag, which has no ApoE, that's the baseline. Stick the flag tagged ApoE in, and these proteins start to decrease. The NLS, it gets even lower in some cases for emerin in particular. And then the export signal causes an increase in LBR and emerin. So this is really one of their bases is for claiming that ApoE may disrupt lamin-associated domains in the nuclear envelope. So next they looked at trimethylhistone, and they wanted to see what that looked like in the nucleus. And that's often bound to the nuclear envelope. Excuse me. And so in the ApoE positive cells, you can see that there's not much there. And when they knock out ApoE, there's a lot more bound to the nuclear envelope. And so here's the densitometry plot. This is in those flag tagged cells. And so they see the same thing, that with the NLS, there's a lot less of the trimethylhistone signal. And then by putting the NES, it gets a little bit higher again. You can see those 2 right here. So this certainly points to an observation that ApoE is somehow involved in aging and disrupting the nuclear envelope. So next, they did a lot of sequencing experiments. I'm not going to cover most of those. They do things like DAM-ID, and then they have a TAC-seq. They wanted to look at heterochromatin accessibility. And so the accessibility actually declines with the ApoE knockout. So they now are saying essentially that lamina association right here is causing a conformational change in this DNA. So one of the final experiments that they did was silence ApoE. And so they took those same cells that I showed in the very beginning, and they used short hairpin ApoE. And in the replicative senescence human MPCs, there's actually a decrease in ApoE, as you would suspect. Same thing for the HDPS, the Werner syndrome, and then the primary human MPCs. So this is just from one old patient, elderly patient. And so they looked at nuclear morphology or in size. And what happened was that it was essentially attenuated from this aging phenotype that you see up here in all cases. So the last piece of data that I'm actually showing for this paper is that in the very beginning, in the abstract as well, they hypothesized that ApoE facilitates nuclear lamina degradation through CAP1, and that this facilitates lysosomal degradation of the nuclear envelope, and it disrupts heterochromatin. So I think this was their attempt at trying to say essentially that there is some sort of role involved here with ApoE. It's not just bound to the nuclear envelope and maybe destabilizing it. So this was their attempt at mechanism. And so what they found in human embryonic kidney cells, but not in the MPCs, is that there's co-IP, so an interaction between LC3, which is involved in autophagy, with ApoE. And so some imaging from their ApoE positive MPCs, they show that LAMP2, another protein involved in autophagy, is actually localized around the nuclear envelope. And so they make this claim then that ApoE is somehow involved in degrading the nuclear envelope and destabilizing heterochromatin. And so the conclusions from this paper, ApoE is increased during cellular senescence, and that ApoE may alter the lamina-associated domains and certainly the nuclear morphology, and that this destabilization may be solely from ApoE. And with that, I'd be happy to take any questions or any comments.
And Max, thank you. That was great. I'm sorry if I missed this. I'm actually commuting from the med school back to campus. But can you comment on what the genotype of the samples were that were used in this study? You made a good point about whether a person is 22, 33, 44, or some sort of het in that. And so maybe particularly for the knockdown experiments, that could be impactful.
Yeah, they actually did not report genotypes for the primary cells that they used, or also the MPCs, which I think would have been very important for this study. Well, if they did, I didn't see it at least. So there may be differences in association with ApoE to the nuclear envelope based off of these genotypes. However, if they blindly went and did these experiments, the likelihood of them having at least 1,3 copy is pretty high. So we see here. But certainly these ApoE isoforms would make a difference.
We can open it up to questions from other participants.
Hey, Max.
Yeah.
So ApoE4 often is thought to act like a null version and lacking activity. And Boo's group has shown that both E4 and the knockout animal have reduced lifespan. What do you... I mean, this work almost suggests that increased ApoE is a bad thing, where the other work would suggest not. Do they discuss it? How would you interpret that?
Well, that's a complicated question, and I hear this well, I see this a lot in literature, is ApoE bad or good? And so far, my impression, I don't have a great answer. My impression is that ApoE is not bad. It seems to be localizing to things to help. So one comment made in this paper in particular is that ApoE goes to the nuclear envelope, and they put a line in there saying that maybe it causes disruption of lipids, which I don't find to be true. If anything, it might be trying to replenish cholesterol stores there. What's interesting, though, is I'm sure these are probably 3,3s, and they are going to the envelope and causing some sort of disruption. I mean, maybe is this a compensatory mechanism trying to help something out? If you remove ApoE, does maybe the other thing get removed with it? Is it bound to ApoE? Because we see amyloid bound to ApoE in our mass spectrometry too. You wouldn't assume that removing, I guess removing those two in tandem would be bad. Anyways, I'm not so certain how to answer that question, Christian, but I would say it's not bad.
If I was wondering if maybe you could comment on how your work, particularly in this domain, maybe is informed by this work, or maybe from a personal standpoint, how you think about that based on what your studies are.
Yeah, so I have a lot of studies all involving ApoE. My primary focus is looking at antioxidant control. One of the things that I currently am seeing is that there are differences in ApoE isoform for baseline levels of antioxidants, which is certainly interesting. More related to this project, Tuck has a fascination with evolution, which many of you know. And we're looking at the ApoE gene cluster, and I wonder maybe is there some regulation of ApoE protein versus the cluster itself. So a paper we published 2 years ago show that there is concerted expression of several gene neighbors of ApoE, and so that this idea that ApoE is the largest heritable risk factor may not be the entire case. There may be a set of genes expressed all in tandem, and that ApoE may be elucidating, or not elucidating, causing the largest effects, but it may be a suite of genes, not just ApoE. In terms of actual interest in nuclear ApoE, I am very interested in it. I've actually worked on replicating Thien de Cara's paper, the ApoE is a transcription factor, and so that's something that is ongoing, and hopefully we'll have a paper out soon on that. But it seems to be something relevant to Alzheimer's disease. That's all I can really share about that.
Any other questions? All right, Max, thank you again. I really appreciate it. And hopefully everyone else will join on at our next USC-Buck Nathan Schock Center Journal Club. Thanks, everyone.